Good morning, everyone. Welcome to our webinar series on weed science. Today, we're going to have a talk from Dr. Neve about gene drive and how it may be applied to weed management. Dr. Neve is the head of the Crop Health and IPM at Agriculture and Horticultural Development Board in London. So, Dr. Neve, thank you very much for your willingness to speak with us, and we are looking forward to hear your presentation.
Thank you, Luis. So I will now take over control of the screen. All right. Okay. Bear with me. So I assume everyone can hear me okay and is now able to see my screen?
Yeah.
Great. Okay, well, yeah, great. Thanks, Luis, for giving me this opportunity. And I'm going to spend the next sort of probably 45 minutes or so talking about some ideas about how gene drive, what might be the potential opportunities and challenges associated with using gene drive for agricultural weed control. I should say that, you know, most of what I'm going to talk about is really just a discussion and comes from a review paper which I wrote a couple of years ago to ask these questions about the potential replications of gene drive. I'll be straight up front and admit that I'm not presenting any data, haven't actually done any research in this area, but I just found, you know, a couple of years ago when I became interested in the idea, the concept, found that, you know, people were starting to talk about using gene drive for weed control, people probably from other disciplines and people who had started to use gene drive in, for example, control of insect vectors that vector human disease. And in some of those papers, you know, the idea was turning up that, for example, gene drive could be used for agricultural pest control, including weeds. So really what I want to do is almost use my perspective as someone who's not a molecular geneticist, who's interested in ecology and evolution and population biology of weeds and how we use that knowledge to develop and optimize weed control. So, trying to go to my next slide and it's not happening quite yet. I'm not sure why that is. Oh, here we go. Okay, so just to start out with a little bit of background, a little bit of context, and I mean this has a slightly UK and European feel in terms of some of the specifics, but I think you'll all, you know, people listening from South America and other parts of the world will recognise the general message here that we need new weed control, weed management tools. Obviously, we've been and continue to be heavily dependent on herbicides for weed control. Particularly in Europe, we're losing a lot of our plant protection products, herbicides, insecticides and fungicides, because regulation is becoming more and more stringent. There's many concerns with off-target impacts of pesticide use in agriculture, be they on biodiversity or ecosystem services or water quality. There's also concerns with the human health impacts of pesticides. And I don't want to today to spend too much time considering the pros and cons of or the sort of scientific basis of some of those concerns. Really, whatever we think about the science behind some of those claims, the fact is that we're losing access any of these tools, and we're also losing access to pesticides because of mounting problems with pesticide resistance and in the context of weeds with herbicide resistance, and all of that together with some technical challenges of trying to find chemistry which can selectively control plant species in a plant crop. All of these things are also slowing the pipeline for herbicide discovery. So it's clear that whilst herbicides will remain an important tool for weed management, we need to think about developing new tools and new approaches for managing weeds in an integrated fashion. So in terms of looking at innovation in weed management, I often think about this as sort of being a pipeline, if you like, where or a continuum of approaches, where one of those is just around optimizing and improving the tools that we currently have available. That includes herbicides, but it's also about thinking about design of crop rotations, cropping systems which minimize the impacts of weeds and maximize our potential to control weeds. So as I say, this is very much about optimizing the tools which are currently available in any given system. There's then a second strand of this, which I've talked about in terms of enhancement of weed management. So that might be looking at the development of new herbicide technology. We've seen many new technologies, or many technologies developed in Australia around the idea of harvest weed seed control. So there's perhaps also the potential to adapt those systems for other agroecosystems, be they in North or South America or Europe or any other part of the world. And then also sort of potential for enhancing approaches based on allele chemistry or based on crop weed competition, on breeding to increase the competitiveness of crops, for example. And then the final sort of part of this picture, and the one I guess I want to focus on today in talking about gene drive, is around the development of new or disruptive technology for weed control. So obviously back 20 years ago, within this disruptive technology, we might have put GM herbicide resistant crops, which have now in most parts of the world become a norm in weed management. But I think looking forward, there are really two areas in which we might see disruption in the future, and that's around sort of the capacity or new techniques related to new methods of what I'm going to call genetic control of weeds, be that RNAi technology or gene editing and gene drive, which I'll talk in much more detail about, but also possibly new opportunities around precision weed management driven by access to more data, artificial intelligence, machine learning, and perhaps new robotics applications for precision weed management. So as I say, I just wanted to sort of set out that as a framework, if you like, for thinking about how we may innovate in weed management, and talk about very much about gene drive at the end of that spectrum, which is about developing new disruptive technology. So perhaps this is, and again, I'm not going to dwell too much on this slide. This is just another way of looking at that weed management toolbox. We often think in terms of looking at the different stages of the weed life cycle through emergence and recruitment, through the seedling stage to adult plants, seed set, then the production of new seeds, which is returned to the soil seed bank. And we have various tools along that pipeline that I just showed previously, which can be used for control of weeds at all of those stages. The key really is that we're trying to develop integrated weed management, which integrates chemistry, genetics, cultural weed management, physical weed management, and biological approaches. And what I want to talk about today is an aspect of genetic control of weed populations. So in thinking about genetic control of pests, and again, I'm talking about pests here, but specifically we're thinking about weeds in the context of this talk, I consider, we can consider genetic control of pests as either direct or indirect genetic control. And I just want to spend a couple of minutes distinguishing between those two things. So when I talk about indirect genetic control, I'm basically talking about genetic control which depends on the modification of crop germplasm, and that can be transgenic modification, but it doesn't have to be. We're just talking about any type of genetic modification, conventional breeding or genetic or transgenic approaches. So it's a form of genetic control which relies on modification of the crop germplasm to impact on plant pest interactions, be that either for disease resistance, for example, so breeding new hosts, new disease resistance into crops, resistance or tolerance to insect damage, or, for example, herbicide resistance. So of course, when we think about weeds, we can think about the sort of classic example of indirect genetic control of of weeds has been achieved through the development of herbicide tolerant crops. So genetic modification of the crop to enhance our capacity to control weeds within that crop. But what I want to spend the rest of the talk, where we talk about gene drive, is really to think more about direct genetic control. And that's where the genetic modification which we're aiming to achieve is direct modification of the pest genome to introduce and proliferate fitness reducing genes into the genome of the actual pest weed that we're trying to manage. So I want to just sort of return to a very little bit of theory here, just in terms of introducing what I'm sure is familiar to you all, the sort of central dogma of molecular biology. So DNA is transcribed into messenger RNA, which is then translated into protein. Now, when we think about using herbicides to control weeds, we're really acting at this end of that spectrum. So we're using chemistry, synthetic herbicides, synthetic chemistry, that interact with plant proteins, enzymes, to limit, slow or stop the essential metabolic processes. So if you like, this is the type of weed control that we're all familiar with, or mostly familiar with in terms of herbicidal control, which is inhibition of essential metabolic processes through interactions of herbicides with enzymes or proteins. We can also think about, and I'm not going to talk about this at any length at all today, in fact this is the only time I'm going to mention it, but we can also think about interfering with the translation of messenger RNA into protein using RNAi technology. So RNAi is a naturally occurring process that limits the expression of target genes through the action of small interfering RNA, which prevent the translation of that messenger RNA. And again, we don't want to go into it in any detail today, but some of you may be familiar with sort of attempts within industry to develop this RNA technology for weed and pest control. What we're talking about when we talk about gene editing, and then as an extension of that, gene drive, is right at the other, is sort of at the other end. So it's about technologies which enable the precise editing of the genome of a target organism to result in heritable changes to gene structure, function, and or expression. Okay, so and that's what we're talking when we talk about gene drive. So I now want to sort of move on to enliven this a little bit with a few examples of direct genetic control which has been used. And in the first instances, I'm going to talk about the use of direct genetic control for insect control. Probably the first development of this idea was this sterile insect technique, which is really just a form of biological insect control. And the idea here is to mass rear insects, and this has largely been talked about in terms of controlling vectors of human disease, so malaria and mosquitoes, for example. So we're talking here about the mass rearing of those insects in special facilities, and then the irradiation of those insects to create sterile males, which are then released into the environment, mate with wild, the released males compete with the native males and to mate with females, but the progeny produced from those matings, because the males are sterile, are infertile. Okay, so this is somewhat similar to inundative biological control. It's a one-off treatment, and I suppose the important thing in the title here is it's a self-limiting mechanism. It's a one-off treatment where you can introduce these sterile insects into a population, and because the sterile males mate with females in the wild populations to produce sterile offspring or sterile infertile eggs, you can drive down population in that way. I'm just going to check because everyone can still hear okay, because I get no signal at this end.
Yeah, everybody can hear.
So if you like, that's an example and one of the first examples of using a method to directly genetically control wild populations of pests. And it relies on it relies on mass rearing of those insects in special facilities and irradiation to make the males sterile. The next development of that technology was something called the release of insects carrying dominant lethal or RIDL technology. And here, this is a GM approach. So this is a transgenic approach where a lethal dominant transgene is introduced into, in this case, it's mosquitoes in most examples. But that lethal gene can be repressed by the presence of tetracycline in the environment. So essentially what happens is that transgene construct is introduced into the mosquitoes in the presence of tetracycline. So in the presence of tetracycline, the gene is not lethal. So in a production unit, in the presence of tetracycline, you can produce thousands, millions of those insects. Those insects are then released into the environment without where tetracycline isn't present, and therefore that gene becomes lethal. Those adult mosquitoes that are released mate with wild population, and all of the progeny from those matings don't survive because of the presence of that lethal gene. So that's sort of the next development of these self-limiting systems is this RIDL, which relies on transgenic technology. And this is being used in, this technology is being developed for commercial application or for use in real situations by a company in the UK that came out of the University of Cambridge called Oxitec. And actually, and I don't know how familiar this company is to some of you, but Oxitec now have a presence, or quite a large presence, in Brazil, and much of their activity in Brazil is around looking at using this technology to control Aedes aegypti, so the mosquito that vectors Zika virus and dengue fever. And again, if any of you are interested, you can go onto their website, and I've just taken a snapshot here of some of their sort of news stories where they claim the success of this technology in driving down mosquito populations which are vectoring. I must say, there's so you'll see at the one of the stories in the left there, they're claiming 81% suppression of wild Aedes aegypti with this technology. I think there's now some, if you do a quick Google search and read some news, there's also now some claims that, some of these mosquitoes that are supposedly carrying these lethal genes are actually surviving in the wild. So yeah, really interesting ideas and being used to control major human diseases in real world situations, and specifically in Brazil. So that's, if you like, where in terms of being used in the field, that's where this idea of direct genetic control has got to. And as I say, the thing with this is it's a bit like what we would call inundative biological control, in that you can release these modified insects into the environment in a one-off situation. They'll reduce the population level by having these fatal matings, or these matings which lead to infertile offspring. So you get some measure of population suppression, but it doesn't last. The population can rapidly build up again. It's a self-limiting system. So what would be really great, what would be the sort of holy grail, I guess, is if you could develop some sort of self-sustaining mechanisms of genetic control. And that would be a bit like the idea behind more classical biological control, where you release some biological agent into wild populations, and you hope that it builds up, becomes self-sustaining, and therefore provides long-term control of those populations. And I guess this idea of self-sustaining direct genetic control is just the equivalent of that. So obviously the fundamental challenge here is that you're trying to introduce some sort of gene which limits or reduces the fitness of wild populations into those populations and to spread through those populations. And obviously that's that's not going to happen through normal Mendelian genetics and inheritance, because anything with a fitness penalty won't spread through wild populations. But within nature, there are various examples of these mechanisms, often termed meiotic drive. And this is a form of naturally occurring intragenomic conflict where one or more genetic loci is able to affect biased or non-Mendelian inheritance of one or more alleles. So effectively, these are naturally occurring mechanisms through which you can get non-Mendelian or non-Mendelian inheritance, which favour the rapid proliferation of certain alleles through wild population. As I say, there's various examples, and I've just shown a few there. I'm not going to go into any detail, but some examples there of this natural mechanisms of meiotic drive in plants, in insects, and also in pathogens. So this shows that, you know, these mechanisms through which you can sort of almost overcome Mendelian inheritance do exist in nature. I mean, they're rare, but they do exist. And so I guess the question then becomes, you know, can we adapt those, or can we look at can we use those mechanisms of meiotic drive or you know, mechanisms which subvert normal Mendelian inheritance to drive fitness reducing genes through pest populations? And so the story sort of develops from there after the discovery of these naturally occurring meiotic drives to the discovery, and I think these discoveries were made, I'm not entirely sure, I think, you know, it was over 20 years ago that these homing endonuclease genes were discovered. And the graphic sort of shows how they act. So these homing endonuclease genes, they're a form of they're a type of selfish genetic element. The homing endonuclease is able to, they're inserted into a gene, normally in an intron, in a gene, in a hosting gene. If you can hopefully see my cursor, so they're inserted here into the hosting gene. They produce these homing endonucleases, which then are able to find the same hosting gene on the homologous chromosome. They cleave, they cut the homologous chromosome in the same place, and then through recombination or through DNA repair mechanisms, they basically insert that homing endonuclease gene into the homologous chromosome. So you've now got proliferation of that homing endonuclease gene through the genome. So, Luis, I'm going to try at this point. I now want to move on, sort of having spoke about some of the, you know, the history of use of direct genetic control, and started to talk about meiotic drive and these actual naturally occurring gene drives. I want to now talk, for the rest of the talk, sort of switch gear and talk specifically about how we might use these gene drives for agricultural weed management. Now, I'm going to try, and I think Luis is going to help me here because we had some issues running it from my computer. I'm going to just try and run a 2-minute video, which just gives a very brief introduction to the concept, much more elegant than I could probably give in 2 minutes of the basic concept of how these gene drives work. And it's a video that was at Harvard University. So Luis, are you able to take control and run that?
Yeah. Can you see that?
Yeah, I can see it. Yeah, I can see. Yeah.
Historically, scientists have been able to alter the traits of domesticated plants and animals, but have not been able to do this to wild populations. Here we use mosquitoes as an example to explain why most genome alterations designed by humans don't persist in nature, and how a recently proposed technology could change that. The transgenic mosquito, tinted blue, has an altered gene inserted into one of its chromosomes. When it mates with the wild-type mosquito, each parent contributes one copy of each chromosome to their offspring. Thus, only 50% of offspring will carry the altered gene, while the other half will inherit the wild-type version from both parents. Even if the altered gene doesn't reduce the likelihood of each mosquito surviving and reproducing, it may persist at a low frequency in the ocean of wild mosquitoes, or it might go extinct after several generations of especially unlucky inheritance. This process is what keeps us from altering wild mosquitoes to prevent them from transmitting diseases such as malaria or dengue. A team led by Kevin Esvelt at the Wyss Institute, the Harvard Medical School, and the Harvard School of Public Health has now outlined a way to build gene drives that can improve the odds that almost any altered gene will be inherited, potentially allowing them to spread through even wild populations. The proposal relies on the CRISPR-Cas9 system, a new genome editing technology co-developed by the same researchers at the Wyss. Gene drive mosquitoes carry both the altered gene, the genes for the Cas9 enzyme, and several guide RNAs that tell it where to cut. When passed to offspring, the guide RNAs direct Cas9 to cut the wild-type version of the gene inherited from the wild-type parent. The cell then copies both the altered gene and the drive when it repairs the damage. Because the mosquito now has two identical copies, one on each chromosome, all of its offspring will inherit the alteration in the gene drive. This same process will be repeated in subsequent generations, causing the alteration in the gene drive to spread through the population.
Okay, so thanks, Luis. I'm going to try now and take back control of the screen. So let me just confirm to me you can see my screen again now.
Yes.
Yeah. Okay, and you can hear me. Okay. So I mean, as I say, that very short video, probably more elegant, probably explains the sort of premise behind synthetic gene drive more elegantly than I could have done in a couple of minutes. So hopefully that was useful as a sort of introduction to the concept. So as I say, you know, there's been a lot of discussion about how these gene drives could be used in nature. This figure here really shows a whole range of potential application domains. You know, and basically in the sentence, they talk about gene drives as new tools for ecology, because it's sort of the first tangible mechanism we have in order to genetically modify wild populations and spread those modifications through those populations. So there's three areas which have generally been looked at. I mean, human health has been the one that's probably motivated some of the first use cases in terms of the control of vector-borne disease. Some interesting and quite topical other applications in human health, also around thinking about immunizing animal reservoirs of disease. Also some thoughts about their use in the environment, particularly in terms of, you know, perhaps introducing beneficial genes into threatened species or deleterious genes into invasive species. But of course, the sort of application we're most interested in here within agriculture for agricultural pest management. As I say, as someone generally interested in novel weed control technology, became quite interested in this also because it interested me, because it's not just a sort of molecular genetic challenge in terms of how do you do this in the lab. It's also very much a population biology challenge in terms of, okay, if you can create these edited wild organisms, how possible would it be, and over what time frame, to actually spread those genes through wild populations? So I wrote this paper a couple of years ago that was published in Pest Management Science, which really addressed that question. How, yeah, what might be the use of gene drive systems for agricultural weed? Again, I'm just going to, hopefully the video sort of explained the general principle here. I just want to explain it again. It's using CRISPR-Cas9 technology, the whole gene drive idea. And it's about, if you see the figure on the left, it's about using gene editing to insert these modified genes, or these cassettes, into wild organisms, and that's Cas9, which is sort of the machinery which cuts the genome at predetermined sites, a guide RNA which guides the Cas9 to whereabouts in the genome to cut, and the payload gene is the gene that you might want to either modify or introduce, or even introduce into the genome. The idea is that you introduce those, and then into wild populations, and then the Cas9 and guide RNA finds the homologous site. So when you get crossing between non-edited and an edited individual, you get that's heterozygous initially for the gene drive, but then the Cas9 and guide RNA finds the homologous chromosome, cuts it, and introduces the gene drive element into the other. So the organism effectively becomes homozygous for that edited gene, and then that's able to proliferate through population. So I think, broadly speaking, that there are three sets of challenges which would need to be overcome in order to make gene drive a reality for control of agricultural weeds. And those are the molecular genetic challenges and constraints. They are the more ecological and evolutionary challenges and constraints, perhaps population biology. How do you actually, given that you can do this in the lab and produce these edited organisms, how would you actually then spread them through wild populations in a quick enough time to achieve effective control? And I guess the third level, and perhaps the one I'm going to spend less time thinking about, I mean, clearly we're talking about a technology here where you can effectively introduce genes into wild populations which could ultimately drive those populations extinct. Now, I think, you might argue for organisms such as mosquitoes which are vectoring human disease, or for weeds which are having huge impacts on crop yield, that driving those to extinction may not be such a bad thing. I'm not sure I think we should be seeking to drive any organism to extinction, but clearly, just generally, this has the potential to be a hugely controversial technology, because, as I say, effectively you can introduce these edited genes into wild populations and at least in principle drive those populations extinct. So I want to just start by, and as I say, this is not an area, I'm not a molecular geneticist, so this is probably the area where I have least insight in terms of what might be possible, and a full understanding of the challenges and constraints, but I'll give you my view as I understand it. I mean, clearly the first of those is, if we want to adapt this technology for use in weed species, we need to have a stable transformation system for those species, and we also probably need to have access to a genome sequence for those weed species, so that we're able to design gene drives which are specific to the species that we're aiming to target. I mean, even now, for I don't think there's any. Someone may correct me here, but I don't think there's any stable. I don't think there's any weed species for which we have well-established stable transformation systems and a genome sequence. Although I think there are many labs working towards that end. So I don't necessarily see that being a fatal limiting factor in the future. There are also some other sort of things which will limit the effectiveness of the gene drive around the homing efficiency. So how efficient is the CRISPR Cas9 and the guide RNA in actually finding and locating the second copy of that chromosome, or the second cutting site? And I mean, it's been shown in other organisms that it's not 100%, and obviously the lower the efficiency of that process, the less effective the gene drive will be. I think because you're trying to find a very specific site, you're trying to design those guide RNAs to find a very specific site in the genome. There's potential for very large and polyploid weed genomes that your guide RNA may guide you to more than a single site in the genome where the genome is very large. And then the next one I need to spend a couple of minutes just explaining as best I can. If you see the figure here, and again, can you see my cursor?
Yeah.
Yeah. Okay. So when we get a break, when the CRISPR-Cas9 and the guide RNA result in a break in this homologous chromosome, there are two mechanisms by which that break can be repaired. One of which is called non-homologous end joining, and that's just basically where the two ends of the broken chromosome are joined together. And the other is homologous recombination, where the second chromosome is used as a template to repair the broken chromosome. And gene drive only works because what you essentially need is you need this situation where you end up with two copies of the drive, so that the organism, the target weed, becomes homozygous for that drive. That's the whole basis on which gene drive works. So you only get that situation where you get this, where the break, DNA break, is repaired by homologous recombination. And again, some of my friends and colleagues who are weed molecular biologists may know more about this than I do, but I don't think we have a good understanding for many of the weed species we work with, what the percent, how much this one happens, the non-homologous end joining versus the homologous recombination. But it only works, you only get proliferation of that gene drive in this situation. And I know from work done in insects, it's been shown that the percentage between those two different repair pathways varies quite a lot between species, between life history stages, between tissues, cell types and organs. So again, that may be one of the limitations, or one of the things that would have to be overcome, ensuring that most of the repair takes place via this mechanism. And of course, we're all of us who work with herbicides and herbicide resistance are aware of, there's many ways, and I'm not going to go into them specifically, in which organisms could evolve resistance to this gene drive as it was being driven through populations. So I think that's sort of a snapshot of all of the potential molecular genetic challenges and constraints. But I want to, accepting that those can be overcome, I want to move to an area that I'm perhaps a little more comfortable with, which is talking about, well, how might we use this in nature to achieve control of weed populations? And I'm going to distinguish between two types of gene drive, one of which we call a population suppression gene drive, and one of which is called a sensitizing gene drive. So my first slide just specifically talks about this population suppression. And the basis of the population suppression gene drive is trying to either introduce a new gene or modify an existing gene, which would effectively reduce the fitness of the weed population in an agricultural setting. So I've got 4 examples here. There could be many more. I'm going to go through 4 examples where I think this could be achieved. And the first of those might be, for example, by the inclusion of dwarfing genes. So we know a fair amount about this RHT1 locus, which can lead to dwarfing. So, for example, would it be possible to introduce genes into weed species which would reduce their stature and therefore reduce their competitiveness versus crops? Another example, and something again which we have some understanding of the genetic basis of seed shattering. So would it be possible to introduce genes into wild populations which led to, for some species where we get seed shattering prior to crop harvest, which limits, for example, the option of harvest weed seed control? Would it be possible to introduce genes which reduce seed dispersal? Another example, some of the major weeds that are dealt with, certainly in North and South America, the Amaranthus species are dioecious. So would it be possible to drive genes through wild populations which might alter the sex ratio in dioecious species to lead to an overproduction of one or other sex, the two sexes. And the final example I wanted to use in terms of population suppression is looking at, would it be possible, for example, and again, these are hypothetical examples, but would it be possible to target gamete production? So perhaps reduce pollen production or even prevent pollen production and therefore limit the capacity of the species to reproduce. So, I mean, as I say, these are all just hypothetical examples of how we might use a gene drive. My own feeling is, it would be, I think it's going to remain very hard to convince any regulator to allow the introduction of genes into wild populations, which could ultimately, particularly if you look at something which reduced or prevented pollen production, which could ultimately lead to the extinction of that species in the wild. Whether that would happen in reality, I very much doubt for a number of reasons, but I think just the very fact that it could happen in theory would mean there'd be some very high regulatory hurdles to jump over in terms of introducing genes for population suppression. I think the approach that might be more palatable is something called sensitizing gene drives, and that's where you introduce a change into a weed population, which would then make it more susceptible to some sort of subsequent management. So effectively, without that management, you might not be impacting the fitness of that organism at all, or that weed at all. So you wouldn't, and that would mean you weren't necessarily introducing something which could lead to that species being driven to extinction. You're just basically introducing something which makes that species easier to control. So I've got a couple of examples here. I mean, one may just be, certainly for target site resistance, where we have a very good understanding of the mutations, often single nucleotide polymorphisms, single amino acid changes, which lead to resistance. It's quite conceivable that we could design gene drives which would return populations to herbicide sensitivity. I would argue, in terms of sort of, really the argument there could be that all you're really doing is returning populations to their wild type state before resistance was selected. So there's not even any argument based on introducing foreign genes into organisms. You're just really using the gene drive to return populations to susceptibility to herbicides. And the other example I've used is perhaps it relates also a bit to one of the examples I used in the population suppression example. But for example, could you, I mean, the big problem we have with blackgrass, or one of the problems we have with managing blackgrass in the UK, is that the seed all shatters and falls back to the seed bank before crop harvest. So we have very little capacity, for example, for harvest weed seed control. So could you think about introducing genes which would limit or increase seed retention, limit dispersal, and therefore increase the potential to use harvest weed seed control? And again, as I say, these are just quite speculative, but just giving you an example of how these gene drives can work in practice. And as I say, I think these sensitizing gene drives are much more likely to be palatable than anything which has the potential in its own right to limit or suppress population. I've got three or four more slides to finish up. I think one of the major challenges in terms of using gene drive to deliver effective weed management in the field is the time it would take those introduced genes to drive through or to propagate through wild populations to reach a high enough frequency to enable effective control of those populations. So this is just some modelling work that was done, not on weeds, not on any specific organism, just sort of some generic modelling work which asked the question, how long would it take gene drives to propagate through populations? Important thing, and I'll come back to this, some of the important assumptions they made was that they were looking at the initial frequency of the gene drive being 1 in 10,000. So that basically means that whatever the population size that you're trying to manage, you would need to introduce gene edited individuals at a ratio of 1 in 10,000, and I'll come back to that. And they looked at various selection coefficients, the negative coefficient actually means that the gene drive would be beneficial, would increase the fitness of the population. Obviously, if you're increasing something, if you're introducing something which increases fitness, it's going to spread through populations more quickly than if you're introducing something that reduces fitness. And they also looked, I talked about this homing efficiency, they also looked at various homing efficiencies, 80% efficiency, 99. I mean, the general message here, for, you know, if you're trying to spread an adaptation which gives a fitness advantage with a high conversion efficiency, they showed you could probably drive that to fixation in a population over 8 to 10 generations. But more realistically, you're really talking sort of 10 to 20 generations when the trait or when the gene is introduced at 10 to the -4, or 1 in 10,000 individuals of the total population. So I mean, you know, that immediately raises a question. You know, we deal with, most of the weed species we deal with have 1 generation per year. So, you know, there's an immediate problem here, I think, in terms of, you know, it may take 10 to 20 years to spread edited genes through populations to the level where they would enable effective control of populations, given that we're looking for sort of 90–95% control or higher generally. But of course, maybe they could be used in more integrated ways, and so therefore perhaps driving these changes through at lower frequency might also have some benefit for weed management. So I think, you know, there are a very large number of quite real challenges in terms of turning this technology into something that would be effective in a short time frame in agricultural fields. So I'm just going to run through some of those challenges and constraints. So we talked on the last slide about, you know, release rates. How quickly something will spread through the population depends at what frequency you're able to deliver it into that population. The higher frequency, the more quickly it will spread through a population. Now, a quick back of the envelope calculation tells us that, you know, let's take Palmer amaranth, where we know, you know, individual plants can produce hundreds of thousands, if not millions of seeds. It's probably not unrealistic to say in a badly infested Palmer amaranth field, though, you know, it's probably a conservative estimate to say there will be 1,000 seeds per meter squared in the seed bank, and that means that for every hectare you've got to get my maths right here, I think it's 10 million seeds per hectare. So, you know, immediately you've got to introduce a lot of edited organisms in. If you're talking about 100 hectare field, you're talking about billions of seeds which you want to drive that gene drive through, and you've probably got to, in order to get effective control quickly, you've probably got to introduce millions of edited organisms into that field. So there's an immediate constraint, I think. You've also got to be sure that the gene drive effectively spreads through a population. It just doesn't become very localized within the population. It spreads right through the population. I mean, I think there are ways to do that. You could think about the way you might introduce these edited weed species into a field would be you'd sow it with the crop. So you would be ensuring that you sowed the edited individuals throughout the field. I think that's another thing to think about. I've already said that there are, you know, we're trying to spread, we might often be trying to spread things through populations which have a high fitness cost. So things that have a cost will inherently spread through populations more slowly because of that cost. You may also find that, you know, that there'll be assortative mating. So you'll get, unless you're able to introduce a local population, an edited local population into your fields, you may find that you get some sort of basically biased mating between the introduced. The native population is more inclined to reproduce with itself than it is with the introduced population or individuals. breeding system, and again, I'll come back to that in the next slide or two. This only works because of the nature of the gene drive. It only works in outcrossing species. You need, in order to drive the gene through a population, you need to have an outcrossing species. Another real major constraint is that you've got there's a seed bank, and in order to really spread this drive through populations, you've got to spread it through the seed bank as well. So there's a sort of natural buffer whereby only plants which you can't drive the edited genes through seeds in the seed bank. You need growing, reproducing plants, and obviously where you've got a seed bank, that considerably slows down your capacity to spread that drive through the whole population. And finally, as I said before, we've talked about the fact that some simulations have shown it will take up to 10 to 20 generations, and we're dealing with species with a single generation per year. So again, this is gonna slow the rate at which we can spread these drives through wild population. So one of the things we did as a result of the paper I showed you that I published a couple of years ago was sort of look at some of what might be the major criteria for identifying what would be the best candidates for gene drive, and I've done that by looking at some of the sort of most widely dispersed herbicide-resistant weeds globally, and we looked at mating system, things about did we have a genome or was one in progress? Are they widely distributed? So could gene drives for these species have application throughout the globe or in multiple cropping systems? And so we effectively looked at that for a number of key species, and what we found was that essentially, as I said before, it will only work in species which are out crossing. So that was the major criteria. If the species was selfing, gene drive won't work. And then we sort of found, based on resistance risk, distribution and availability of genome, sort of the prior, we found that according to the analysis we carried out there, that Lolium rigidum was perhaps the number one candidate, just given its wide distribution and some of the other traits there. So, as I say, my feeling, and I had, for various reasons, including the fact that I've now moved to a different job, and one of the colleagues that I was going to work on this work moved before I did. We felt that the next stage, before we even went into the lab or conducted lab experiments or lab work to establish the feasibility of editing weeds and setting up gene drive systems, we felt there was really a modelling exercise here to ask, well, can we actually achieve effective weed control through gene drive in realistic timeframes by considering all of these factors which would impact on that? We didn't do it, but someone got to it before us, or at least addressing some of those questions. So this was a study that came out of Australia, from CSIRO in Australia, published last year, where they looked at modelling gene drive spread through weed populations with seed banks, and they effectively look. The V is basically the annual germination fraction. So a V of 1 says that all of the seed in the seed bank will germinate in the current year, and therefore there is no seed bank, through to they went through to V equals 0.2, which meant you only get 20% emergence per year, or 20% germination per year. And what you'll see is, again, if we take the sort of middling, with an annual germination of about 60%, they were saying that gene drive would lead to the fixation of edited genes in populations over about a 20 generations. And again, for annual weeds, that means 20 years. So again, we come back to that question about, how could we actually, and the other thing to say here is their introduction, that the introduction rate they were using was far higher than the 1 in 10,000 that was used previously. It's now slipped my mind. It's quite an important. I think they looked at something like 1 in 200. You would be introducing 1 in 200, and that's a huge rate of introduction. So of course, I said I wasn't going to dwell on this. I mean, there's been lots of discussion. hugely powerful these gene drives could be for a whole range of applications. But there's a huge debate going on about, you know, the ethics and the regulation about this. We, you know, we can alter entire species, but should we? So I'm going to draw to a close now. So just putting that out there is obviously a really important part of this discussion. So just to bring this to some conclusions, yeah, we started out by saying there is a pressing need for innovation in agricultural weed management. I don't think anyone disagrees with that. Gene drive systems provide a really intriguing possibility for the direct genetic control of weed populations, a completely new paradigm, if you like, for how we could think about managing weed populations. So really interesting and really worthy of further study and investigation. Again, spoken from someone who very rarely did or does set foot in a lab. You know, according to my reading of this, you know, the molecular genetics is challenging, but likely feasible with enough resources thrown at it to enable gene editing and gene drive within weed species. To my mind, the greater difficulties just in terms of the practical application are associated with the population biology, and how would you actually spread gene drives through weed populations rapidly enough to affect, you know, effective control? Would farmers really commit to a strategy where they might have to introduce these edited individuals into fields and wait 10 or 20 years, 20 or 20 generations to get appreciable control? Maybe they would. But you know, that's obviously going to, I'd imagine, impact the way people think about this technology and adoption. Only possible in outcrossing species, and of course, I didn't touch on it very much here, but there's huge regulatory, ethical, and societal implications. But as I said, I think this idea of sensitizing drives may be a way to get round some of that in the future. So final slide, just to sum it all up, you know, to my mind, this is really interesting technology. You know, I've given a proceed with caution green light on the molecular genetics. I think, you know, the ongoing advances in synthetic molecular and genomic biology applied to weeds increasingly should make the technology possible. I've given the sort of amber light, if you like, to the ethics and regulation. I think currently the regulation is restrictive and wouldn't allow this, but there's many efforts to sort of safeguard gene drive technology, and I think, you know, that suppression should actually say sensitizing. I think, you know, in future, you know, with the right safeguarding and possibly with these sensitizing rather than suppression drives, it may become possible. But I think the real limitation that we need to address is around the population biology. How do we spread gene drives through wild populations rapidly enough to make them effective? And you know, without wishing to sort of finish on a downer, I think that is the major challenge, and I think it's also makes it really interesting because it's an area where I think clearly molecular geneticists and population biologists would have to work together right from the outset to ensure that what's achievable in the lab is actually practical in the field. And with that, I will finish and invite any questions.
Yeah, there are two questions there, if you want to read, or...
Yeah, let me stop sharing now and then. So, I mean, the second one, perhaps I'll address first. So it says, I don't know if everyone can see these, but what is your advice to the next generation of weed scientists in terms of research priorities and needs to make gene drive a reality? I mean, I think we really need to do those modeling studies in order to understand, yeah, we need to understand what we expect gene drive to deliver and over what time course in the field. And I think the only way to really do that presently is through some modelling studies, and then allow the modelling studies to sort of drive the attempts to make to develop the technology for use in the field. So I think that would be my advice, is that, you know, I think, yeah, perhaps there might be a temptation to go off and develop this technology in the lab, because I think we probably can. But I think it's really important to also ask the question, how would it work in the field, and how can we develop it in a way that it will work in a reasonable time frame in the field? And there's another question. It says, is it possible that natural systems of mutation correction interfere with the establishment of gene drive? Yeah, I'm not. Just need to think about that one. Yeah, I mean, I'd invite, if Nelson wants to sort of expand on that. I mean, I think what I would say as a general answer to that is, you know, I think these gene drives will be a huge selection pressure associated with these gene drives, particularly if you're trying to drive fitness reducing genes into populations. And I think that there'll always be a variety of mechanisms which could evolve to overcome or subvert those gene drives. But I don't know if that's that's a sort of bit of a woolly answer to that question. Nelson wants to.
Yeah, I think so. Yeah. I think we have no questions. Any other questions? All right, Paul. Thank you very much for your. Oh, there is more there? No. Oh, there is another one there.
Yeah. Yeah, I mean, I think that's an interesting question. And I think that is potentially another way to think about this. But you know, so it's basically, could a plant virus be engineered to deliver the gene drive system? I think certainly there are some systems which could be used to use plant viruses to introduce changes, to introduce perhaps more transient changes in genomes, which wouldn't necessarily be inherited from year to year. So they would be more equivalent to the sort of self-limiting systems that I talked about for insect control originally. But yeah, I think there's an interesting question there, which wouldn't I don't think those viruses, unless there may be things I'm missing, but I don't think they would enable modifications to be driven through populations generation to generation. But I certainly think you could use viruses to deliver some sort of genetic modification within a season or within a generation.
You have another two. Maybe you can answer these other two, and we can adjourn.
So I'm just reading. Yeah, I mean, I think the one from Edwin, you know, acknowledges the fact that, you know, it acknowledges another inherent difficulty with gene drives, in that we're trying to drive something through a population which makes that population less fit. You know, so on the one hand, that's going to slow down the rate at which it spreads through populations. And it's also, you know, going to potentially lead to greater pressure for, you know, the gene drive to be overcome through evolution of some sort of resistance to that gene drive. So yeah, I think Edwin's question addresses you know, another fundamental difficulty. I think one of the you know, the whole premise of gene drive is it can be used to drive fitness reducing genes through populations, but the more the fitness reduction, the slower they'll be to spread.
Mm-hmm.
And then Diana, could gene drives have any probable mechanism to select resistance? Again, so I might have to ask Diana to tell me if I misinterpret the question, or I mean, is the question here related to, you know, could we use gene drives to introduce resistance into populations?
I think she meant that if the nature will find a way to be resistant to the gene drive.
Yeah, yeah, okay. Yes, I think yes. Yeah, it may be that, you know, for example, one way in which you could see that resistance evolving to the gene drive is if the sequence that the CRISPR-Cas9 is being directed to mutates in some way that you no longer get recognition of that sequence, that cutting point, then that's an obvious way in which resistance could evolve and the gene drive would no longer work.
Mm-hmm. Yeah. I think that's the last one. Paul, thank you very much for your seminar. It was excellent. It was very good to see you again, and I wish you and your family to be healthy and safe. And we really appreciate your time. Thank you.
Well, again, thank you, Luis, for the introduction. I've just realised my camera was off. I don't know if it was during the whole. I couldn't. Yeah. Anyway. All right.
Thank you.
Okay. All right. Thanks, Luis, and thanks everyone. Bye.
Thank you. Bye.
Bye.